3, 2, 1, go.
Good afternoon, everybody. My name is Philip Ahern. I'm an assistant professor at the Lerner Research Institute at Cleveland Clinic, and it's my pleasure on behalf of the Early Immunology Committee to host today's seminar. And to remind everybody, our mission here is to promote and highlight early stage investigators who are doing really outstanding work in the area of immunology. It's my pleasure today to introduce Dr. Sophie Acton, who is soon to be associate professor at University College London at the MRC Laboratory for Molecular and Cellular Biology. So Sophie started off her academic career with a master's in pharmacology from University of Bath. Following that, she moved to the lab of Eric Sahai at University College London, where she performed for graduate studies. And then she obtained a highly competitive Sir Henry Wellcome postdoctoral fellowship, which she completed initially in the lab of Shannon Turley at the Dana-Farber Institute at Harvard, and then returned to the UK to work with Kitano Reza-Sosa. And I think Sophie's been doing some really, really elegant work, both as a postdoc and since she established her own group in 2016, looking at the interplay between dendritic cells and fibroblastic reticular cells in lymph node function. And I think incorporating lots of different kinds of approaches to try to understand how the cells in the lymph node and the immune cells that are infiltrating the lymph node are interacting to regulate their function. So with that, I want to welcome Sophie, and just to remind you that if you have any questions, please place them in the chat. So Sophie, it's all yours. Thank you.
Thank you so much for the introduction and for the invitation. I'm very pleased to tell you about some of our recent work. So I am really a cell biologist and interested in cell interactions, cell communication, how tissues then coordinate cellular behavior. We happen to ask those questions in the immune system. So I'm an immunologist on one hand, but a cell biologist at heart. So our understanding of how adaptive immunity works will be very different than perhaps a classically trained immunologist, but I think we've asked different questions and brought to the field some different ways of thinking about how we understand how immune responses are regulated. And today I'm going to show you some of the background and talk you through some of our very recent work on how lymphoid tissue remodeling is controlled at a mechanical or biophysical level and what the impacts are on the adaptive immune response. So our model system has been the lymph node, but many of our secondary lymphoid structures have these same basic map and structure to them. So they construct two main functions. Firstly, as we think of them as meeting points for immune cells, we need antigen presenting cells to meet and activate our lymphocytes. Our lymphocytes need a space in order to proliferate, differentiate, and develop the responses and effector functions that we need them to do to then give us a systemic effective response. But the second function, which is perhaps their original function that we may sometimes overlook, is that they are also, at the same time as being meeting points, they're also really important in how we balance fluid. So lymph flow from peripheral tissues will filter back through our system, trickling through structures in our lymph nodes in order to not only balance fluid, but also give those signals and help with our scanning of our entire body through these structures. So as a tissue, they're really interesting. They have many different inputs, many different outputs. They're exceedingly complex. And yet they are robust. They manage to remodel every time that we have an adaptive immune response. And we continue to have effective adaptive immune responses throughout our lifetimes, given some aging. But these tissues have this robust feature that their structure remains intact and remains functional over and over again through cycles of immune responses and immune challenges. So here is a mouse lymph node, and I'm showing you an image that has no immune cells in it at all. Here we're imaging the stromal architecture of the lymph node. The larger vessels you can see with the basement membrane labeled in collagen, with collagen 4. These are the blood vessels that move through the lymph node. These are the entry points for lymphocytes from the systemic circulation. And all of this meshwork that sits between the vessels are a fibroblastic reticular cell network made of mesenchymal cells, tightly joined together, and a continuous network and tightly ensheathing extracellular matrix, which forms the conduit, which will filter fluid through the system. And you can see that even without looking at any immune cells, this space is really busy, really organized. And these stromal cells are firstly organizing different tissue niches. So broadly, our T cells and B cells require different signals and therefore sit in different areas of the tissue. In the mouse lymph node, the B cell follicles are towards the periphery and the T cell zone is in the middle. In larger human lymph nodes, the same structure is true, but you get many repeated patterns through the tissue in the same structure. So beyond being structural, though, these are providing growth factors, survival signals, chemokines in order to organize this structure, but also to give the lymphocytes exactly the right niche, all the signals that they need in order for that adaptive immune response to be productive. So we've been very interested in understanding how immune responses are regulated by the stromal structures that support them. And in particular, looking at these fibroblastic reticular networks and how they facilitate immune responses. So to go back in time and how I started being interested in this question, when I started my postdoc with Dr. Shannon Turley, who now is at Genentech, but this work was done at Dana-Farber at Harvard, I took some experience I had in cell migration for the first time into immunology. To that point, I hadn't really studied immunology beyond a few courses in my undergraduate. And we asked the question about how is it really possible that a dendritic cell becomes activated in a peripheral site and could follow a chemokine gradient across meters, so many, many multiples of the cell's actual size. How is that possible that a chemokine gradient is the signal that brings in antigen-presenting cells to the lymph nodes? It's just too much distance. The differential gradients that people had suggested just perhaps didn't quite make sense. Perhaps there was more going on. And at the same time, the field had become very interested in these stromal structures. There was a couple of key papers showing that these stromal cells were not just architecturally, structurally important, but actually were producing a lot of these key molecules. So we started to ask what the interactions were between dendritic cells particularly and the fibroblastic stroma of lymphoid tissues. And what we found was that even in the absence of chemokine, so once we removed them from the lymph node, chemokine expression was very low. But even in the absence of chemokine or blocking chemokine, the dendritic cells were very able to walk in any direction across these stromal networks. And we were able to find that the interaction between a C-type lectin, which is upregulated on dendritic cells as they become activated, and a glycoprotein podoplanin, which is expressed at high levels on the surface of these fibroblastic reticular cells. This interaction was able to activate the dendritic cells' cytoskeletal signaling processes to activate RAC and actin polymerization and allow these cells to spread and move along these networks. So adding an extra layer to our understanding of how antigen-presenting cells actually get from peripheral tissues and arrive and move around within our secondary lymphoid organs. But beyond that, and moving on, we actually very interestingly found that when dendritic cells were crawling along these networks of fibroblastic stroma, the stromal cells themselves seemed to be responding as they were in contact with these cells. So there was a reciprocal signaling, not just they were presenting a ligand for dendritic cell migration, but these same protein interactions was actually also modulating the cytoskeleton of the fibroblastic reticular cells. And why might that be important? Well, having seen the complicated structure of the lymph node, we started to ask, well, how is that structure remodeled to, as we all know, lymph nodes expand really quite rapidly. The first thing your doctor might see if you were unwell, they might feel for your lymph nodes, and they will be swollen if you're undergoing any kind of immune response. You need more space. So we're jumping from this simple observation that dendritic cells, when they interact, will produce more protrusions on the fibroblasts that they're walking on. We hypothesized that actually this may have something to do with lymph node remodeling as a whole, and actually lymph node expansion. And that turned out to be the case. So for long story short, this paper is also published several years ago now. But if we do an immunization model and watch how lymph nodes expand, if we immunize mice that do not express any of this C-type lectin, CLEC2 on their dendritic cells, this is deleted, driven by CD11C, lymph nodes just don't expand to the same degree as their control counterparts. And moreover, we can replace that signal of the CLEC2 using a recombinant protein injected as you would an adjuvant to a vaccine, give them more of that, just one signal. And not only can we rescue that defect in lymph node expansion, we can actually make wild type lymph nodes really, really big. So this was a very important paper at the time, and it made us understand that dendritic cells had this really important dual function. They're bringing antigen and they're activating our T cells, but actually without them, we wouldn't also be able to start that process of priming and remodeling the tissue. And this became something that I've worked on further as I began my own group at University College London. So we asked some somewhat basic but unknown questions as we began to delve into this understanding of adaptive immunity at a whole organ level. And what we asked was, firstly, how do you begin to expand? We had some ideas about the fibroblasts stretching and growing and changing their cytoskeleton, but we didn't really understand all of the processes or how that would work. But beyond that, we know that when immune responses are ongoing, you do actually get the fibroblastic reticular network proliferating. And how is that controlled? What were the triggers for that genuine growth of the tissue? And something that we're still working on today, which is a much more tricky problem to get at. We would also really like to understand how tissues that have so vastly remodeled and grown and generated all this space can undo all of that remodeling and revert back to their steady state. How do you resolve an immune reaction in order that your lymph node doesn't keep growing or stay at this larger size forever with too many fibroblasts, too many blood vessels, as all your lymphocytes have now left? So I'll talk you through some of what we have discovered along the way. We don't fully understand resolution, but we found out quite a lot of interesting information along the way about how the mechanics of this system control the remodeling of the cellular networks. So most of the work that I'm going to present to you next was done by a very talented PhD student, now Dr. Harry Horsnell, who has since left the lab to undertake a postdoc in Australia in the lab of Scott Muller. I'm sure he will go on to do many great things. He came in enthusiastic to ask this question, taking on novel methods that we hadn't investigated before, very keen on the biophysics, and with collaborators we undertook to understand what mechanical forces the stromal cells and the T cells would be under during adaptive immune responses. So broadly, we split our understanding into extrinsic versus intrinsic changes, meaning that these networks of fibroblasts would be under pressure. We hypothesized because just the movement of lymphocytes in and out of this system, there must be some balance of forces in order that the lymph node size can remain constant. And then that balance may shift during an immune response in order that there can be some additional space for T cells. And a mechanical explanation for remodeling did make some sense to us. If you consider something like a rag knockout mouse that lacks lymphocytes, their lymph nodes are then extremely small. So the structural properties of the lymph node really seem to be fixed for the number of T cells that are there. They're going to make the structure around the requirements of the lymphocytes. On the other hand, cells can certainly feel force on an individual level. They can be stretched, they can be squashed, they can feel hydrostatic pressure. They have adhesion to the underlying matrix, which can give them a feedback. And these are also things that we wanted to investigate and how they would change through the course of immune responses. So as a starting point, we had a really close look at the fibroblasts in the network, which when I'd first entered into this field just wasn't really possible. I'm going to pause the movie here and explain what we're seeing. So in the magenta is an antibody stain for perlecan, which is an extracellular matrix component, which is on the conduit networks. These are these fine bundled fibers of extracellular matrix, which filter the conduit, the fluids coming through from the peripheral tissues through the lymph node space. And in cyan, we've stained for filamentous actin. So all of the circles you can see around, these are the T cells packed in, and this is their tight cortical actin. You will mostly see actin in a T cell around the cell cortex. But if you notice along the perlecan here, these cyan filaments here are F-actin cables, which if I play on further are also decorated with phosphorylated myosin light chain, meaning that they are indeed contractile. And these filaments are along the outer edges of the fibroblastic reticular cells as they're stretched along these networks of extracellular matrix. And all of that is contained within the cell membrane of these fibroblastic reticular cells. We can do this on a whole organ level. We can zoom right in and have a look at these structures all over, and we would see many of these repeated units. So it wasn't that there was contractile structures in particular areas, that in steady state, most of our FRCs looked like this. And the fact that we have even an actin cable at all, like bridging across this area of cell membrane, implied to us that there was indeed tension through this network, even in the steady state, balancing out some forces from T cells. So where would the forces come from in steady state, and how would they change through lymph node expansion and immune activation? For this, we did some very simple data analysis on the images that we could take in on 3D sections. So the T cells in the T cell zone is where we focused our attention. And that's when the majority of the fibroblastic reticular network is in a mouse lymph node at least. And we quantified how many T cells were squashed into a defined space. And here is presented in 100 micron squared areas. And you can see that as we immunize and we take time points shortly afterwards, this is a day 3 and a day 5. So this very acute beginning phase of lymph node expansion, the number of T cells packed into the same physical area is increasing pretty rapidly and remaining higher up to day 5. We also quantified the spaces between these fibroblastic fibers of the network. And this is an algorithm we had used previously in our first story published as my postdoc and lymph node expansion. We generated an algorithm where we fit circles into the spaces in the network of decreasing size, and then we define the space by that array of circles as a histogram. So if we've got lots of larger spaces, we'll see a shift in the population, and we can then do statistical analysis to show that the spaces are larger or smaller. And again, we can see that over the course of these early phases following immunization, as the lymph node is getting bigger, the spaces in the network are also getting bigger. So we've got more T cells packed in, but they're also packed in a stretched network, also implying that there is force being felt by the tissue. But in order to really prove there are forces and tension in this tissue, the only way to do that really is to take biophysical approaches in live tissue and to measure forces. And for this, we collaborated with a lab of Yan-Lan Mao and with Eva Polu, who is now working at the University of Cambridge. when we discussed the possibility of doing techniques such as laser ablation, which is where we were going to effectively zap and break some cellular structure that we would image live. And then if it pinged back like an elastic band, we can imply from the recoil distance how much tension was over that cell junction or through that area that we've ablated. However, in order to do that, we needed a mouse model where we could live image our fibroblastic reticular network we need all of the cells labeled, we need to see all of the detail. And although there were some models available to image fibroblastic reticular cells, we really wanted a membrane marker in order to see all of the detail of this intricate network. So we generated our own new mouse model, which we're really pleased with. It uses PDGF receptor alpha to drive the membrane GFP expression, and also a tamoxifen inducible Cree, which will become important later, as then we can genetically also manipulate these cells. But the first thing we did with this mouse is to use this live GFP signal. Ex vivo, we take lymph nodes and keep them alive in thick fibrotome slices. Now we can see the network in steady state. We can zoom in, and then we can really speed up our imaging to the image many, many times a minute per second. And we turn up our laser power. And if this plays, we were able to show you how we can ablate this network. And you'll notice that both sides of the ablation site are recoiling and moving apart. And if we quantify this over time, over many, many ablations, we're able to plot data, as you can see on the left, where we can plot the distance of before and after over time and get this recoil curve, which we can then calculate tension in the network. And so in steady state, we are now able to say for the first time that, yes, indeed, the fibroblastic reticular network is under tension, which means that for the lymph node to stay in a steady state size, the tension in the network is buffering forces from the external environment, which are the T cells packed into those spaces. We can also now with this system ask how the mechanical forces in the tissue change over time. So following immunization, this is now at day 3, we know those spaces are becoming stretched. You can perhaps also see that in this image. We knew that there's actually more T cells packed into the same area. So we were in fact expecting that tension in the network would be higher. Whereas if you'll see the data, actually tension at this point is lower, which was a surprise to us at the time. But in a couple of slides' time, I will explain to you how this was possible. If we take a later time point now, this is at day 5, and zoom in and we zap, we laser ablate this area, you can actually see quite a lot of recoil here. And day 5, consistently tension was at higher than steady state levels, which we can quantify here. So this decrease at day 3 was rather mysterious initially, but the day 5 is perhaps as we expected. More T cells pushing in, stretching the network. Those cells are feeling that stretch by being under more tension. The next question we had to really address is where's that tension being held? What structures are responsible? Because as I showed at the beginning, these fibroblastic reticular cells are tightly associated with extracellular matrix. And when we're doing these ablations, we're really ablating both. There's no way to physically separate the two. But at day 5, you'll see here, we had previously undertaken some work to understand how this conduit network was remodeled during lymph node expansion, and then found, and we see again here, that actually during expansion, the extracellular matrix is not excessively remodeled and rebuilt. It actually snaps in places and becomes somewhat disrupted. So at day 5, we do definitely have cells plus ECM, but we also have areas where the extracellular matrix has become under so much force that it has in fact broken. And it's the cell that bridges this gap, which you can see in this image here. Here we've used the same mouse model crossed to a confetti reporter line so that we can image individual fibroblastic reticular cells in the network. So we can see this one cell here rendered in yellow, bridging this gap of broken extracellular matrix. And here the matrix is imaged using a second harmonic signal. So this is really looking at our fibrillar collagens. So this led us to really investigate the cell as the source of our tension. And if we'd seen those strong actin-myosin cables also in our imaging, so we asked whether actin-myosin contractility was responsible for the tension through this network. And we can test that by treating with a Rho kinase inhibitor. And in steady state, this really decreased tension when we do our laser ablation experiments. At day 3, it made very little difference because the tension was already lower, but this high tension we were seeing at 5 days post-immunization was reduced really to baseline with just this actin-myosin inhibition. So it led us to conclude that really most of the tension that we see increased in this system is coming through the cells resisting the tension through their own cytoskeleton. So to reverse slightly and try and explain, why does the tension in the tissue reduce, in fact, at day 3, when everything else is telling us it should be, the lymph node is bigger, the cells are packed tighter, the network is stretched. So why is physical tension in the tissue actually lower? And this is why it was important to actually measure it. So we hypothesized that actually it must be the cells themselves intrinsically adapting in order to make more space. So now they're not responding directly to the T cells, but at this phase at day 3, they've been primed in order to actually physically relax, and they're doing that beyond the need. So we're actually overshooting. But to address this experimentally, what Harry did was to use a laser trap, optical tweezers, you could also call this method, where we trap a bead in a laser and we attach it to the cell, live imaging in vitro, and that way you can pull on the membrane of the cell and test how stiff the cortex is, how much tension there is in the membrane. Because if these cells are going to stretch and make more space, and the same number of cells are extending their protrusions, then actually you need more membrane, you need to become more flexible. So this is what these type of assays look like. The bead is trapped in the center of the cell. You can see it on this target. And as it pulls a tether of membrane from the cell, and we're really measuring the attachment of the membrane to the underlying cortex here, you'll see that that bead moves out of the center point of that laser trap until the tether pulls itself off and releases. And again, using the disturbance, the distortion of that bead out of the center, we can calculate effective the membrane tension of the cell. So now we have a way of measuring that. And if you remember at the beginning, I had shown you we do have one pathway that we know very well changes the actin cytoskeleton regulation of both the dendritic cell and also the fibroblastic reticular cell. And that's that interaction between the C-type lectin, COLEC2, and the glycoprotein podoplanin. So this was what we tested first, and indeed it really changes the intrinsic mechanics of individual FRCs. So when we treat cells, pre-treat now with a couple of hours with COLEC2, the effective membrane tension of the cells is dramatically lowered by more than 50%. But it's also extremely rapid. So we can measure the same cells before and after COLEC2 treatment, just taking 10-15 minutes between measurements. And we can see even in that short time frame, the same cell, almost the same area, will be much lower effective tension. And this is because downstream of podoplanin, we have regulation of the esrin, rhodixin, and myosin family, which link the membrane to the actin cortex. And these proteins, the phosphorylation of these proteins is much lower following COLEC2. It's also transient, interestingly. So we wanted to really understand that this was definitely through this protein-protein interaction that we had identified. But it's possible, because we're using COLEC2 recombinant protein as an FC fusion protein, that actually this could be an effect through the FC, it could be binding something else on the surface. So we undertook to take a set of controls to prove that it really was specific. And here we took fibroblastic reticular cells where we have genetically knocked out podoplanin, and then we've reintroduced either wild type or mutant podoplanin in order to test this. So the wild type reintroduced, now using a construct, responds similarly. So COLEC2 treatment reduces membrane tension. if we introduce a mutant of podoplanin, this is a threonine mutant to alanine in the cytoplasmic region, this cannot bind to CLEC2. Now it becomes non-responsive and the CLEC2 doesn't do anything. And the other mutant we tested, we changed the serines in the cytoplasmic tail of podoplanin also to alanine, so it can no longer downstream signal. And again, it now becomes non-responsive to CLEC2. So we're really confident that whether CLEC2 has other effects on the cell, this effect that we're measuring is specific to this protein interaction with podoplanin. So what would that mean for the cells and how they might respond? So we hypothesized that if you need to get longer, structured, larger distance, you're going to need more membrane and you're going to need to generate a larger cell volume really quite quickly. By flow cytometry, we can see that fibroblastic reticular cells at this phase are slightly larger. But we tested this directly using a simple assay where we use a hypertonic solution and force water into the cells. We can measure how quickly they swell, and they will eventually burst. But we can then get a really simple measurement, in fact, of how quickly and how much membrane they have available in order to expand. And when we pre-treat the cells with CLEC2, that swelling that we induce with the hypertonic solution is much more rapid, and they can swell to a larger size than control cells. So we also investigated where this additional membrane might come from, and our conclusion was that they probably have membrane stores in the form of caveolin, and these might well be also mechanically sensitive. Potentially, we should investigate this further to see if this has anything to do with sensing the changing mechanics in the system. We also were able to look in the tissue at what these cells look like, how their morphology did change on an individual cell level in the tissue. So again, we've used our mouse model to induce the confetti reporter, and then we searched through the tissue to find two cells next to each other with differential labels. So now we can really investigate how cells interact with one another. And in the control, we can see cells joining and binding to each other. They don't join end to end, as you might see in some diagrams in review articles, but they actually overlap to a regional amount. And then the protrusions that they form seem to form almost feet, which kind of then spread out onto their neighboring cell. But we did see some interesting changes following immunization, where they seem to interdigitate with one another much more than they do in the control setting. And there are many, many more protrusions on each cell, some of which reach out to neighbors, some of which follow other paths in the extracellular matrix, and some which are just waving around in the air. So there are other mechanisms at play here which we haven't fully investigated in order that these cells can make additional protrusions, and that's work that's ongoing. So, but our original question and why we were studying mechanics of the system in the first place was to try and test whether FRC proliferation and tissue growth was really triggered in a mechanically sensitive way. So we'd chosen these time points following immunization because really in steady state, FRCs are very quiescent. They're barely turning over. They're not really in the cell cycle. Very few of them are. But at day 5 following immunization with this protocol, they're just beginning to enter the cell cycle and begin to proliferate. And our first investigations were really, and other people in the field have asked the same, is there a progenitor population? Is there an area of the lymph node that lots more FRCs are going to come from? People had suggested that the marginal reticular cells, which sit underneath the capsule, could be more proliferative or more stem-like. There were also suggestions that the fibroblastic reticular cells that sit around blood vessels, like a pericyte population, they may also be the source of additional cells. But we saw no evidence for either of these claims when we looked. So the beginnings of proliferation, and we just look for cells by Ki67 staining, so cells that are actively in cell cycle, then it's completely stochastic. It's all over the lymph node, but very much at random and fairly evenly spread, which I think gave us further evidence that the trigger for entering the cell cycle really probably was mechanical rather than a chemical source or a progenitor population. But we investigated this more fully also in vitro. So indeed, FRC proliferation is very mechanically sensitive. We can do simple assays in a dish or on collagen gels in order to test this. So on very rigid surfaces, so plating these cells on glass, they will proliferate much more rapidly than if we plate them onto a very soft substrate. And here we've taken different stiffnesses, ranging from super soft all the way up to glass. Whereas if we change the two mechanically sensitive pathways that we now know are relevant for FRCs in the tissue, which is actomyosin contractility, which we used in our laser ablation experiments, or podoplanin, which we know is affecting the intrinsic mechanics of the cell and the membrane tension. If we compare knockout FRCs for podoplanin or ROCK inhibitor treated FRCs, we now reduce not just the total level of proliferation, but they're now not so sensitive to this changing in their environmental stiffness. And as an aside, interestingly, podoplanin expression itself is mechanically sensitive. So there's a feedback loop here that when we put cells into very stiff substrates, they will very rapidly upregulate podoplanin in response to that stiffness. And again, that can be blocked if we block the feedback through actomyosin. And here we've treated them with a ROCK inhibitor and we can prevent the upregulation of podoplanin. So not only is podoplanin conferring mechanical sensitivity to the FRCs in order to change the way they proliferate in vitro and probably in vivo, in fact, its own expression is mechanically sensitive. So having thought we had that nicely tidied up, the reviewers said, well, you really have to show this in vivo, which was a big comment, but to be fair, it is a fair comment, and it added a huge amount to our understanding of the system by doing this set of experiments. So we were asked to delete podoplanin in vivo and show what difference it made to the mechanics of the tissue during expansion. We had a collaborator in Birmingham who had generated a podoplanin floxed mouse, and we used our own CRE with the GFP tag and the inducible CRE in order to delete that in vivo. And here you can see our control cells. Sorry, the labels here are missing. The podoplanin is in magenta across the GFP tag network. And when we delete podoplanin by addition of tamoxifen, we can get rid of almost all of the podoplanin expression in most areas of the lymph node, which we can also quantify here by flow cytometry. So we're getting a good shift by a log or so reduction, but we're also getting plenty of FRCs that are now completely negative for podoplanin. So we then tested what the effect this had on the mechanics, and we were able to repeat our laser ablations. And in the controls, as we've seen before, we get lower network tension at day 3 and higher tension at day 5. And we weren't totally sure what we were expecting with podoplanin. We knew that it was affecting the intrinsic membrane tension, but in vivo, what would this mean? And actually what we did see is that they become very insensitive to either this lower tension that they should have. They don't now respond to CLEC2 in vivo, so they can't become more stretchy and lower their membrane tension. But they also now don't experience higher tension at day 5. So they've just become unresponsive. And this is quite different than our ROCK inhibitor treatment, but it gives us now 2 mechanical perturbations we can compare in vivo. So the Y27 drug, which inhibits ROCK, will actually effectively mimic our relaxation at day 3. But we now don't get the increased tension at day 5. Whereas podoplanin, they can't reduce membrane tension and tension at day 3 or day 5. But both of these, although they're different, they both affected numbers of fibroblastic reticular cells, which we've quantified GFP positive fibroblastic stroma and made sure that they're CD31 negative. So we're really looking at our FRCs. And at day 5, with either of these perturbations, the number of FRCs that have proliferated and the total population is significantly lower. So mechanical cues in vivo are required in order for FRCs to proliferate. We were also asked then, so what impact that had on the immune response? And so protoplanin deletion actually affects lymph node expansion. With this, completely phenocopies the deletion of CLEC2 in dendritic cells. So perhaps this is what we were expecting. And this constrains numbers of both T cells and B cells. The actomyosin inhibition is actually slightly different. So we're actually allowing the lymph node to expand with the pharmacological means, but we haven't given the right cues. So the lymph node does expand, the FRCs don't proliferate. And what we found is that actually we're then specifically constraining the T cell compartment, and the ratio of T cells to B cells was significantly different in these expanded lymph nodes. If we take this experiment out longer and look at a later time point, so something like day 9, as we've done here, this is when our T cells are really activated and we can measure the beginning of generation of memory cells. Now we can see that if we constrain lymph node expansion and mess with tissue mechanics by deletion of protoplanin, we're actually severely affecting the generation of memory cells. And here we're looking at the T cell compartment. So the rest of this story is published. You can find it online, and look out for Dr. Harry Horsnell, because I'm sure he'll go on to do many other great things. He's an extremely talented scientist. What I'd like to spend the last few minutes discussing is some unpublished data where we've investigated this phenotype more thoroughly, and we found some very interesting additional effects of deleting protoplanin and changing the mechanics of the tissue if we take a longer look. So firstly, I've told you a lot about this particular protoplanin interaction with CLEC2. But actually the contact between a dendritic cell and a fibroblastic reticular cell is having really extensive effects on the signaling pathways of both cell types beyond just these two molecules, or even beyond actomyosin contractility. Here we did an experiment where we differentially labeled our fibroblasts and our dendritic cells and undertook to do mass spectrometry. And we looked for total proteome, but also phosphorylated peptides, so we could get an idea of signaling pathways that were changed. And to just summarize here, there's plenty of interesting things going on. But overall, the take-home message is that contact with DCs gives a huge difference to the phosphorylation of peptides in the FRC. We're really changing signaling pathways. And variance here on the y-axis shows that actually many of these changes are CLEC2 dependent. So this upper population cluster of points is actually our dendritic cells that have no CLEC2 expression. But we also looked at the transcriptome to understand this better. And this actually gave us even more insight into what we thought was a complicated signaling pathway in the first place. Firstly, it's a ligand for CLEC2 and changing dendritic cell migration. Now CLEC2 is a ligand for protoplanin and changing FRC, actomyosin, and how lymph nodes expand. But when we look at the transcriptome downstream of this pathway in the fibroblasts, we found there was a lot more than that going on. So protoplanin expression alone regulates a whole number of genes in a huge range of pathways, which I'll summarize for you, but happy to discuss in detail. And this work is actually now in a preprint if you'd like to see the full details online. So many of these genes on the left are not dependent on CLEC2 binding. They are just regulated by whether the cell expresses protoplanin or not, which is a new idea. We didn't know what protoplanin did. Now we know that it doesn't even need a ligand. And this could be relevant to many settings, because protoplanin is expressed in many cell types, in many tissues, well beyond the lymph node. It's highly upregulated in inflammatory conditions across all different contexts. So they're including cancer, for example. So just expressing protoplanin will change hugely how that cell is signaling and what state it is. But the other thing that we did test is the response to CLEC2. And here we saw in the top panel here, if you look at the first cluster of genes, we see genes that are inhibited in their expression by CLEC2, which phenocopies the loss of protoplanin. And this would be like our actomyosin pathway. So CLEC2 binding to protoplanin allows the cells to stretch and elongate, and knockdown of protoplanin does the same thing. They become less contractile. But if you look further down this list, there are genes that respond in totally different patterns, and actually CLEC2 is also turning on a whole bunch of alternative pathways. So we now consider this key signaling pathway in at least 2 new ways. There are podoplanin-dependent signaling pathways and transcriptional events. And to some degree, when a dendritic cell and CLEC2 is bound to podoplanin, some of these are switched off, but also an alternative pathway of CLEC2-dependent genes downstream podoplanin are switched on. Here I'm showing you some string analysis of some of these genes, and I'll just highlight a couple of things. So the CLEC2, one of the biggest cluster of genes that are turned on are to do with ribosome biogenesis, really telling us that these cells are switching states, switching functions, really waking up and getting ready to do a different phase of function in the lymph node, because now we're responding to an adaptive immune response. So not quiescent, we're not steady state, no more homeostasis, we're ready to react. Whereas if we look at the podoplanin-dependent genes, so this would be in a steady state now, we found large clusters of immune-related genes, which we found particularly interesting, having thought of this network and this signaling pathway as mostly related to actomyosin contractility. Now we need to consider that actually maybe some of these immune regulatory properties that fibroblastic reticular cells have in secondary lymphoid tissues may well be because they express podoplanin, and that's something we investigated further. So indeed, actually, when we delete podoplanin in the lymph nodes, as we did for the mechanics work, but leave these cells a little bit longer to really adjust and not having podoplanin any longer, and in these longer term experiments, but still in steady state, we haven't immunized these mice. We really find that the FRCs have changed phenotype well beyond. They're not just FRCs that now no longer express podoplanin. They're FRCs lacking podoplanin that are also lacking ICAM1, VCAM1, chemokine expression, key molecules such as CD44, tetraspanin, CD9. And so we're actually changing the phenotype of the FRCs themselves really quite substantially. What does this mean for the tissue? So again, this is steady state. We have deleted podoplanin and just waited. We know that we've upset the mechanics of the system, but over time, what does that mean? And first thing you might notice, because the image on the right here is very blue, we get a big upregulation of extracellular matrix deposited. We also see disruption of the network. I think you'll see also, if you look not even closely, there are big holes in this tissue. And this is not because we ripped the tissue cutting it, but actually because the network is starting to fall apart. And I'll show you that in more detail here. So apologies, the labels here are missing, but the left is the control. And here we've got a nice FRC network surrounding blood vessels closely. We've got overlay of our GFP tagged mouse model with the podoplanin staining. In the B cell follicles, then we have much less stroma and more space. This is near the edge of the lymph node. So if we look at the podoplanin deleted mouse, which is here on the right, we've got lots of areas where the network has really separated and pulled apart. And if we look in the B cell follicles, we've got this overgrowth of stroma. It's all GFP and not stained with podoplanin because we have deleted it. And we can quantify these changes to network structure. We attribute these to the problems that these cells now have with their network mechanics and their ability to adapt to the forces of the T cells coming in. So if we just leave these cells longer, they are continually under force and the network falls apart. So we've used a skeletization method where we can quantify branch length, we can quantify kind of networks within the network. And you can see that when we delete podoplanin, we reduce the number of branches, the length of those branches, and also the footprint of these mini networks, although we're taking two-dimensional samples. But perhaps most interestingly, and we don't fully understand this phenotype, we are hugely affecting lymph node compartmentalization. So remember, back to the beginning, our B cells should be sitting neatly in follicles at the edge of the tissue, which you can see here. in our control and stained with B220. They are clustered together densely into B cell follicles and separated physically from the T cells in their paracortex. They are supported by their stromal networks. This all looks very normal, but when we delete protoplanin, now we've got a lot of overgrowth of stroma. There's a lot of GFP in where the areas where the B cell follicles should be, which is not normally there. And actually the B cells have either got up and wandered off, or they're not any longer properly clustered in the follicles. So we're really probably also affecting in this model the phenotype and function of our follicular dendritic cells, which are the specific stroma, also fibroblastic stroma that support B cell follicles. So I will run out of time, but I will stop here. This new work, which, although unpublished, is pre-printed and available on bioRxiv if you'd like to see more. And to summarize, I'd like to thank the team, and just to say that you don't have to be an immunologist to study immunology, and you don't have to understand a technique to give it a go and to branch into other areas. And I really enjoy collaborating with people where they know things and they're actual experts in things I know nothing about. It's one of the most exciting parts of being a scientist. So most of this work was done by Harry Horsnell, who's here, supported by a few different postdocs in the lab. Victor Martinez did the initial work on extracellular matrix, and Dr. Spiros Makros is the one whose preprint I showed you at the end. Thanks also to our funders, Cancer Research UK, the European Research Council, the Medical Research Council here in the UK, and also our European funders who funded various people as they came to do their work with us. Thank you very much.
Thanks a lot, Sophie. That was really, really awesome work. And I think it's kind of an area that enough immunologists probably don't think about these, you know, the CD45 negative compartment of the lymph node is often just dumped, right? Ignored by immunologists. Because
Yes.
obviously it's playing a really, really important role. So there's a couple of questions. So one question I had is regarding these, you had shown there was these protrusions and they seem like the FRCs almost start to touch each other. And then you had also shown that their proliferation seems better on harder surfaces. So I'm just thinking, do those two things fit together? Does like neighbor contact, is that like a signal to proliferate? Like is somehow an assessment that they need to expand the size? Or is there any way that you can determine if those, I mean, it seems really difficult, but if that contacting can somehow modify the function of the FRC?
So normally a cell like a fibroblast would have the opposite. So there's contact inhibition of migration. They'll separate from each other, but they will also know and feed back from each other as the cells get confluent in the dish, but also perhaps in tissues have similar mechanisms. So actually to stop proliferating, FRCs are unlike normal fibroblasts in that, so in a dish, they will overgrow each other and grow almost in layers, which is extremely unusual. So in the tissue, I think anything goes here. They definitely need touch and their physical contact with their neighbors in order to make this network. So they must have mechanisms in place to overcome any inhibition of their behavior, but they're also not mobile. They're not actually migrating. So while we talk about them as fibroblasts, a lot of their behavior is much more like a glial cell, perhaps, where they work as a population, and there's so much we don't understand. I think making new protrusions is a mechanism that we're starting to understand, and Lindsay Millward, who's another PhD student who recently finished in the lab, has a beautiful story that we haven't quite put out yet, but we have some ideas about how they make those protrusions, which was her work.
Awesome. So there's another question in the chat, which is just saying, great talk. For the DC FRC interactions you showed earlier, do you see them in vivo in the lymph nodes? Are you able to visualize that in vivo?
Yes, so there's a lot of resident dendritic cells in a lymph node. So using something like CD11C or MHC class 2 almost the whole FRC network looks decorated with some kind of myeloid cell. So yes, there's a lot of them. To image specifically migratory cells is technically challenging, but some people have done it. There are some papers by other labs that have used CD11C YFP to live image dendritic cells in tissues, and you see very close contact with the stroma.
I had another question. It's probably, this is more conjecture on your part. I guess it's a resolution question, which, you know, I think is the next area that's really missing, I think, in terms of chronic inflammation. But I guess the model, right, is that as the immune cells disappear, that the FRC network might shrink and it resizes. But do you think it could be the other way around or circular, that actually somehow at some point that the FRC network starts to shrink, and then that forces a purging of the lymphocytes because there just isn't like a network for them to stay there anymore?
It could be either. It really could be either. And it's difficult to do those experiments. Our working assumption now is that the T cells and B cells leave into the circulation and go out first. And that's purely from taking lots of fixed time points through immunization studies with different adjuvants. And actually, we generally see a lag of the stroma behind the T cells in whichever direction. So on the way up, lymphocytes expand and FRCs are like, oh, there's not enough of us, now we'll proliferate. And it's the opposite in the other direction. So that data is also unpublished right now, but you see that there's actually a phase where there's too much stroma and not enough lymphocytes, and then they start to reduce the number. We know they must undergo apoptosis, so we've done some experiments doing that, but also capturing that in imaging is very challenging. But our feeling now is that the stroma respond to the requirement of the lymphocytes rather than it being an active squeezing of them out.
Sure. Okay, yeah, that makes sense. I think we have time for one more question. There's one more in the chat, and it's asking whether you know if FRC growth in 3D is more limited than in 2 dimensions.
The proliferation.
They're asking about growth, but I guess in general, probably.
Yeah, I think in general, yes. So in the tissue, we see very little actual proliferation. So I think what we're seeing in vitro is a sped up hyperactivated version because they're in serum, because they're on glass. It gives us a way of measuring differences, but I think, yes, that comment is very much true, that in the tissue, everything is much more controlled.
Okay, awesome. Well, thanks again, Sophie. This is really an outstanding talk and kind of a lot of food for thought, I think, for people. And thanks everybody for joining on the live stream. And don't forget, we'll be back in a couple of weeks. So thanks very much.
Thank you for having me.
So the live stream should end in a second. I think Shu is just going to take care of that.